So we're continuing with our virology lecture. This is the second part. And in this lecture, we're going to focus on specimen collection and diagnosis of viruses. Now, for a viral infection, specimens should be collected very early. So, right when someone knows that they might have an infection, they're starting to get symptoms, that's a good time to collect a specimen. And what specimens are collected is going to vary. Just like with bacteria, it's going to vary on what type of disease the person might have, what types of viruses are suspected, as well as the time of year, because certain viruses are much more common cause of infection at certain times of the year. So some viruses, like your rhinoviruses or cold viruses, are more common in the winter. You'll have some other foodborne viruses that might be more common in the warmer summer months. So all of these come into play when you're thinking about the type of specimen you want to collect. Now, like your bacteria, you can look for viruses in a variety of specimens. So if you have a virus that's causing an upper respiratory infection, you might get a throat or nasopharyngeal specimen. So the nasopharyngeal aspirates would be the better upper respiratory specimen to use to look for a virus. Nasopharyngeal swabs are acceptable to look for certain viruses. Not all viruses would you want to obtain a nasopharyngeal swab for. Throat swabs can be acceptable for other viruses, enteroviruses, adenoviruses, herpes simplex virus. So it really depends on what virus is suspected as to what type of specimen would be acceptable or considered to be the better specimen for that type of infection. Stool specimens, so some of our viruses do cause diarrhea, things like that, so you would expect a stool specimen. So stool and rectal swabs. Stool specimens are preferred over the rectal swabs, and commonly you're going to get a stool specimen to detect rotavirus, some of the enteric adenoviruses, and the enteroviruses in general, because entero means intestines, viruses, these are viruses that cause intestinal infection. Urine, you can detect virus in the urine, and you would want at least 10 mils of a clean catch first morning urine. So first morning urines tend to be more concentrated, and so they might contain more organisms than other urine samples throughout the day after you've been drinking a lot of water and flushing and diluting things out of your body. So you can possibly find different viruses in urine, such as cytomegalovirus, mumps virus, rubella, measles, adenoviruses. You can also get skin and skin lesion samples, and this would commonly be a type of specimen for maybe an enterovirus. You can do something called a zinc smear, and what this is for viruses that cause a pustule, a vesicle. You'd want to what they call unroof the vesicle, so break open the top of that vesicle and then take a slide and press it, make an impression smear of the vessel right on the slide, and then you would do immunofluorescence or possibly electron microscopy or other techniques on that zinc smear. So you could do a zinc smear on herpes simplex virus, which causes a vesicle or pustule to form, or varicella zoster virus, which is the cause of chickenpox, which causes the little pustule lesions that kind of scab over, as well as possibly cytomegalovirus. Serum is probably going to be one of the more common specimens you would get in a clinical microbiology laboratory. So serum is going to be used for all of the serological based tests, which is probably the majority of the types of tests that will be performed for viruses. So you'd want 3 to 5 mils of serum. You're commonly going to use serum to detect antibodies. 2 serum samples are usually collected. You want an acute serum sample right when someone starts getting symptoms. You want to get a serum sample, and then you want to get a convalescent serum sample 2 to 3 weeks after you collect the acute specimen. That's because usually when someone's just infected, they may just be developing antibodies, so the antibody level will be very low. But 2 to 3 weeks after they have had that infection, their antibody levels are going to be very high. So you'll see a 4-fold rise in the antibody titer in the serum, in the convalescent serum compared to the acute serum. And that would mean that someone was recently infected and they are a few weeks into this infection. Now, like some of the other organisms we've talked about with viruses, you really want to process this specimen immediately. These specimens shouldn't be sitting around at room temperature. They should never sit at all any higher than room temperature. Generally, specimens are going to be transported on ice. They need to be refrigerated until they're processed, and they should be processed within 12 to 24 hours of collection. So these samples shouldn't be sitting around in a refrigerator for a week before you do any type of testing on them. You can possibly store certain specimens up to 4 days if they are at 4 degrees of refrigeration temperature. If you are going to store longer than that, so 5 or more days, you then must transfer the specimen to a freezer, either a -20 or a -70 deep freezer. Now, cell culture is one way to grow up and identify your viruses. So unlike bacteria, viruses can't be streaked out onto an agar plate and then stuck in an incubator for a day or two and then look for colonies. That's not going to work with a virus. So what you commonly do, way back, what you would do is inject eggs, and you would grow your virus up inside like chick eggs. But now we have cell cultures, and you can buy many different types of cells. So depending on what virus you want to grow, you have to make sure you are using the correct type of cell, because remember, there's viral tropism. Not all viruses will infect every single cell. So for cell culture, you can purchase specific types of cells from companies like ATCC, the American Type Culture Collection, and you can grow them into these special tissue culture flasks with media. So it's sort of like a broth that you would grow bacteria in, but it's different types of nutrients. The common media that cell culture is grown in are Eagle's minimal essential media, which is usually called like Del Bucco's, Eagle's, DMEM, Earl's balanced salt solution. Usually with this media that you purchase, it has usually they're kind of a pinkish reddish color because they have a pH indicator in them of phenol red. They have a buffering system in them that keeps the media at a neutral pH of around 7.2. So what you want to do is you grow these cell cultures in the special tissue culture tubes with this special media. The media contains fetal bovine serum that you have to add to it, and you also usually are going to add antibiotics to it. That's because viruses aren't killed by antibiotics, but you want to make sure you don't contaminate your cell culture with bacteria. or fungus or yeast. So you use different types of antimicrobial agents so that other organisms, if they were to get in, would get killed, but your virus would be able to grow in the cells. You'd then incubate this cell culture in your typical 35 to 37 degree incubator for up to 1 to 4 weeks, depending on what virus. So just like bacteria, different viruses take different lengths of time to grow. Now, once a virus infects a cell, some viruses cause what's called CPE, or cytopathic effect. Not all viruses do that, but the ones that do, if you were to take a specimen and inoculate a cell culture and that virus infected those cells would start to show symptoms, kind of like you would if you were infected. And different viruses make different symptoms of these infected cells. And there are virologists that have been looking at CPE for many, many years, and they can actually sometimes tell just by looking at the CPE what type of virus is infecting those cells. So we said we have different types of cells that you can actually purchase. Now, a cell line is a culture that has been passed, grown up in that media with your bovine serum, and has been passaged where you take some of those cells out and put them into fresh media. That's called passaging cells or subculturing. You can have primary cell lines. A primary cell line is a cell culture that has been passaged only once or twice. You can have a low passage cell line, and low passage cell lines will viruses will infect some of these low passage cell lines, and low passage are 20 to 50 passages. There's something called continuous cell lines, like Hep2 cells. These types of cell lines, it doesn't matter how many times you passage them. So for low passage cell lines, once you passage them 20 to 50 times, you have to throw them out because viruses will not infect them anymore. But a continuous cell line, you can keep passaging them into fresh media, and viruses will continue to infect those cells. You could also have a blind passage where you passage cells and fluid to a second cell culture tube, and you may not actually know what virus you have in that situation. Now, we mentioned cytopathic effect, and this is one method that you can use to detect virus. So we already said viruses that will infect these cells tend to cause symptoms in the cell. So in other words, the eukaryotic cells that the viruses are infecting in the cell culture will change the cell's morphology. So the cells will do different things. They'll either detach from the surface. So eukaryotic cells tend to stick as a monolayer, one single layer of cells on the surface of tissue culture. Virally infected cells will pull themselves off, and so you'll just have a lot of cells floating around. Some viruses lyse cells, so you'll get a bunch of cells lysing and getting killed. Some viruses cause that Syncytia formation, where it causes all the cells to fuse together and it looks like one big gigantic multinucleated cell. So when you're identifying a virus, you want to look at which cell type is the virus infecting. That's going to give you a clue, or at least let you rule out certain viruses, because viruses do have the tropism. How quickly does that virus produce cytopathic effect, or CPE? because certain viruses take a certain amount of time to cause CPE, and that will give you a clue as to what virus it might be, as well as the morphology of the CPE. Is it causing the eukaryotic cells to turn into these little round cells where they normally are stretched out? Is it causing them to detach from the surface? Is it causing Syncytia formation? That morphology can give you a clue as to what virus it is. So some of the typical CPE that you might see with various types of viruses. So for example, the smallpox virus will cause cells to round up, get round where they're usually more stretched out, and you'll see cytoplasmic inclusions. Herpes simplex virus will cause those cells to fuse in that syncytia formation where you get that giant multinucleated looking cell, and you'll also get inclusions. Adenovirus causes cells to clump together, and you'll see inclusions in the nucleus. Poliovirus doesn't have inclusions, but it will cause cells to lyse. You likely will not look at many cell culture viral infected cells. Most clinical laboratories do not do cell culture and do not identify viruses using cell culture. A few clinical microlabs do, and only for a couple of viruses. So this isn't going to be the main method of identifying a viral infection in a clinical lab, even a clinical lab that has a virology department. So you might grow up one or two viruses in that lab, and you might be able to look at the cytoplasmic effect of those two viruses, but not that common. So here on the left-hand side of this slide, you see a certain type of cell that is not infected with any virus. This is just what it looks like if you just look at these cells growing in a tissue culture flask under the inverted microscope. Now on the right hand side you could see the CPE. These cells are you could tell they're kind of sick. Some of them have died. They're not in a nice monolayer. Some of them are balled up, rounded up, very circular in form. So you can tell that these cells are what would be called sick cells. They're not feeling well. They definitely have symptoms or CPE. Here is typical CPE of the respiratory syncytial virus, and that classically is going to cause that syncytia formation, where you could see right in the middle that clump of cells that looks like one big huge gigantic multinucleated cells. Those cells fuse together. And here's measles virus on the top. Again, uninfected cells. Usually you want a nice uninfected cell culture as a control. Make sure your media is clean. Make sure your cells are healthy. And then you look and compare to your cell culture that has been infected with your virus. And again, these are different cells here. This is measles. These are B95A cells. And you can see there's a big difference between the cells on the bottom of this slide compared to the uninfected cells on the top. Now there's another method that some laboratories do. Again, only those labs that have virology will do some of these cell culture and looking for CPE. And the shell vial culture is a modification of the typical conventional cell culture. The shell vial culture allows you to identify a virus more quickly, so you still have to infect a tissue culture, cell culture, a cell monolayer. You then centrifuge that monolayer down to concentrate it, And then you're going to do an immunofluorescent type of staining on it so that you stain the viral antigens. And then you can look under a fluorescent microscope and see the virally infected cells. So the thing that's nice about shell vial culture, some viruses don't actually generate CPE in infected cells for days, maybe even weeks. With the shell vial culture, it's an immunofluorescent stain, so you can actually identify the virally infected cells long before they actually develop the CPE. So it allows you to make a more rapid diagnosis. And here is cytomegalovirus in a shell vial culture where you can see the fluorescent organisms. Now, here is the most common method that any clinical microbiology laboratory will use to diagnose a viral infection, and that is going to be serology. And actually, serology isn't always done in the microbiology laboratory. So, way back when, I used to work in a hematology laboratory for a couple of years. And in that hematology laboratory I worked in, we did all of the serology. So we did ELISAs, your enzyme-linked immunoabsorbent assays, for many different types of bacterial infections and viral infections. We did CMV, we did some HPV, we also did bacteria, Borrelia, Lyme disease. So serology is sometimes done in its own area of the laboratory, sometimes it's done in micro, sometimes it's done in heme. It depends on what laboratory you happen to be working in. Regardless of that, serology is going to be a much more common method, as I already said, to identify viruses. Now, the problem with serological based tests is you're not actually looking directly for the virus like you are with using CPE or shell vial culture. You are actually looking for the virus. For serology, you're looking for antibodies against a virus. So you're dependent on the host immune response. So sometimes, if an individual hasn't developed an adequate host immune response yet, you can actually test negative when in fact you do actually have an infection. And this is one of the reasons why, for many viruses, you would get a paired sera. You'd get the acute serum right at the beginning when you're just getting the symptoms, and then a convalescent serum would be collected 2 to 3 weeks later. And if you truly are infected and maybe the acute serum didn't show any antibodies, you were negative, the convalescent serum should rise fourfold, which means that you definitely have an acute infection. Also, some of the serological based tests, a lot of your EIA types of tests, will look at IgM and or IgG. So the IgM antibody response in a human is the initial response, and then the IgG response takes over. So if someone has IgM antibodies and not IgG antibodies, it means they've just recently been infected. If someone has IgG, much higher IgG, and very little IgM, it means they've been infected for a longer period of time. They've got a chronic infection. So this can be very helpful for some types of viral infections, knowing if it's an acute or chronic infection. So the problems with why you would, we already talked about some of the problems with the serological based testing is you might not have developed an immune response yet, so you test negative falsely because you actually do have an infection. You're not looking at the actual virus, you're looking at antibodies. But why would you want to do serological based testing? And there's many reasons why serology is the best choice. That's because a lot of viruses are not culturable. You can't keep some viruses, you can't just grow them up in cell culture. So for those types of viral infections, you need to rely on the serological based tests. Also, you can determine a person's immune status. I already said you could determine if someone has an acute infection or a chronic infection based on their IgM and IgG antibodies. You can monitor the immunosuppressed patients. Are they starting to develop any antibodies? Is their immune response kicking in at all? And you can do some epidemiologic and prevalence studies using serologic testing. Now, in a lot of laboratories, especially micro laboratories, they're starting to do molecular based tests or nucleic acid based testing. Most of the nucleic acid based tests in the clinical laboratory are amplification based tests. So PCR or polymerase chain reaction, branch DNA, NASBA or nucleic acid sequence based amplification, as well as microarray. And for PCR, labs are doing general PCR. Most of them are doing real-time PCR. So for those of you that are taking molecular diagnostics or have had some molecular biology before, you already know what these tests are. For those of you that are going to be taking molecular diagnostics and haven't had any molecular, we will be talking about all of these different types of tests in molecular diagnostics. So we're not going to talk about these tests and how to do them, but just know that a lot of viruses, human papillomavirus is one, that there are nucleic acid based testing are becoming the gold standard for some of the viruses. Now, the advantages of nucleic acid tests is you get a diagnosis very quickly. For cell culture, some of these viruses could take a week or more to grow up and generate a CPE, or even grow up in a high enough amount to detect using shell vial, the shell vial culture assay. So with nucleic acids, you can get a diagnosis in a couple of hours. Nucleic acid tests are also very highly sensitive. You can detect those non-culturable viruses just like serology. You can quantitate nucleic acid tests. So you not only can you tell if someone is infected with a virus, you can tell how much they're infected. You can do viral loads. And you can also do nucleic acid tests to look for many viruses at the same time. So if there's a person that has an infection and you don't know what they're infected with, and they may have 10 different types of viruses, you can test for all 10 in the same test. So those are some of the advantages of using nucleic acid based tests. For nucleic acid based tests, you're also looking directly for the virus, just like CPE in the shell vial. Unlike serology, where you're looking for antibodies. So that's another advantage. Now, there are some disadvantages for nucleic acid tests. They usually are more costly. You need some specialized training. So usually in some of the micro labs that are doing molecular, you have to have special training before you can work in that area. So just because you have a micro certificate and you've passed your boards, or just because you're a CLS and you've passed your boards, they likely require molecular specialty or training before they'll let you work in the molecular area of the laboratory. Also, for nucleic acid based tests, you can detect even dead viruses. So that sometimes may not be good because Your body could have fought off the infection. You actually don't have an infection anymore, but you still have some dead viruses there. And since you're testing for the nucleic acids, just because they're dead, you'd still have some nucleic acids there, so that could be a disadvantage. So we're going to start out now in the next part of our lecture talking about all the various viruses. Now, we could spend an entire semester on a virology course and really talk in detail about every virus, but this isn't a virology course. It's more of a bacteriology course that we kind of throw in some other organisms that aren't as commonly tested for in the clinical laboratory, but you need to know about them anyway. So we're going to start out with the DNA viruses. So here's a little schematic on your DNA viruses. You have those that are enveloped and you have those that are non-enveloped. In those that are enveloped, you have your double-stranded DNA viruses. Those that are non-enveloped, you have double-stranded and single-stranded. So we're just going to mention some of the more common viruses within the larger viral group, and we're not going to talk in detail, just in general, what the various viruses are and what diseases they cause. So you're not doing biochemical testing on viruses like you are in bacteria. So there are a lot of viruses. We're not going to talk about every one, but it's a little more simple because you don't have to memorize every single biochemical test in order to diagnose it. Diagnosis is commonly going to be serological based tests where you do an ELISA and you get a titer and you know if someone's positive or negative. So in that sense, viruses can be a little more simple, but they are complex organisms. So we're now going to move on to the third part of this lecture where we will start talking about all the different DNA viruses first, and then we'll move into the RNA viruses.